Welcome back to chapter 3 of HAS4650. In this chapter, okay, we'll talk about some current development and quality assurance issue for herbal cosmetics and cosmeceutical. Okay, so we'll talk about some efficacy and safety, toxicological and stability evaluation and help to control the Chinese medicine in Hong Kong and some current and contemporary development of the cosmetic. So to start with, so there are several sources for consumer on product safety about the aromatic cosmetic or personal care product. Okay, so you in USA or in Europe. Okay. So the information from both of these agencies is readily available from their website. Okay, so you may have a look for example, cosmetic ingredient review. So in most countries, the safety assessment of new finished product is a basic requirement and often to be documented for legal purpose. Okay, so including the product information packaging. Okay, so must be provided. Okay, so according to the regulatory entities. In USA, okay, so of course, I mentioned in chapter 1, so Food and Drug Administration, that is FDA, okay, so issue FD&C Act, okay, so which mandates that the cosmetic manufacturer must substantiate the safety ingredient and products before their market. Okay, so of course, in other European countries, they should follow the EU cosmetic regulations. So let's start with the toxicological evaluation. Okay, so that is the safety assessment of the cosmeceutical. So in fact, we should show the toxicological profile of each ingredient. Okay, so for example, preservatives. Okay, that is the aim is to evaluate the risk of application to healthy human skin under normal and foreseeable use conditions. Okay, of course, we may recruit the target groups. Okay, so in terms of different age, exposure time, frequency, and use conditions. So the evaluation also include the determination whether these ingredients may be systemically available. For example, we may investigate the mechanisms of bioavailability. So, and some factors, for example, physical, chemical factors that affect the properties of the ingredients and the metabolic processes within the skin and possible systemic burden. Okay, so and the assessor requires to evaluate the data about the thermal absorption. Okay, so based on the systemic burden and toxicological profile in order to calculate what we call the margin of safety. Okay, so and we combine these data. Okay, so to estimate and predict the safe use of new products. Okay, whenever there is any new formulation has been established. So that's why we should have some what we call the preliminary safety testing. Okay, it's conducted before we progress to skin tolerance and compatibility testing. Okay, so remember, as I mentioned in chapter 1, so in European country, animal testing is legally forbidden now. Okay, so, and this method has increased the ability to recognize the potential hazards and to identify the concentration of ingredients that are likely not to cause adverse thermal response. Okay, so that's why or we call the preliminary judgment about the tolerability of ingredient. So on our skin, okay, so of course, need to confirm by suitable testing on human. So that's why I would like to introduce a very famous test to do. That is the human patch test. Of course, we recruit the volunteer, okay, who are able to conduct the assessment on their skin tolerance after single application or after repeated application. Human patch tests can be used to evaluate, the irritancy of our skin, whether you're allergic to a certain ingredients. A specific volume of product is then applied to occlusive or semi-occlusive patch. Usually they are applied to the back. After regular or specific period of time, the patch is then removed. Okay, and the skin site is clinically graded for objective irritation, whether they contain the erythema, papules, pustule, okay, or blister, something like that. So, and then a subsequent patch is applied to the same site. Okay, so the common used protocol is used to evaluate the potential for cumulative contact irritancy, allergic sensitization, or potential for phototoxic response, and even the sun-induced allergic sensitization. So, of course, the tests differ in the frequency and number of patch that you applied on the skin and whether the UV exposure is used in the protocol. Okay, so in fact, we have different patch tests and controlled use tolerability tests are also conducted to evaluate other indications. Okay. So after the toxicological analysis, one that is the microbiological testing will be performed. Okay, so to see, so whether the formulation are adequately preserved to ensure the quality over the claimed shelf life and to protect against the contamination during the consumer use. So that's why we will conduct a microbiological challenge test. This procedure has been taught in another module, Has4636, that is the preservative and microbiology. So in the first year. Okay, so of course, the steps we have gone through in that modules. So usually we need product preparation, and then inoculation, incubation, and then we need to count the number of cells. That is how many CFU. We estimate the surviving microbes. So, and then we add the product. Again, we inoculate it and incubate it. So, and then we interpret the results using the microscope. Whether the preservative can access the bacteriostatic agent that inhibit the growth of the bacteria. So we have another term called bactericidal. That is used to kill the bacteria. So and after the microbiological test, we will conduct the stability test. Usually it involves two directions, that is the chemical and physical stability test. So the purpose is to see whether the product meets the intended quality criteria, as well as their functionalities and aesthetics aspect. So the common used test condition for physical chemical properties will be the long-term storage at a controlled room with controlled temperature, so stress testing, or some night exposure test. We also need to test for the packaging materials because packaging material may directly contact with the ingredients inside. So that's why many stability indicating parameters are monitored over time. So that's why it also involves the analytical analysis of the formulation to predict the product shelf life. So why the product can indicate the shelf life to you? So because it undergoes a series, of calculations and the testing to estimate, the product shelf life. So and then So the final cosmeceutical formulation is subject to different testing. So including the testing of key ingredients, stability testing, and as I told you, microbiological challenge testing. Okay, so when the product or formulation pass these tests, okay, so we still need to test on the package design, package engineering, and product development. Okay, to see, okay, to find out the most suitable package material, container, or closure system for the dosage. So that's right. Okay, so all in all, we have three major types of, for example, that is the efficacy testing, stability testing, and safety testing. Okay, so efficacy testing, for example, the skin barrier functional test, so the skin whitening test. Okay, so and moisture content test, so stability, physical or chemical stability test in terms of environmental condition. Safety test, usually we have the toxicological testing, including human patch test or microbiological challenge test. Okay, so why we need to test on the package? Because that ensure to give the functionality, cost, parameter, and package specific marketing claims. So testing can be conducted by yourself. Okay, that is what we call the in-home usage testing. Okay, so it's a required step to test the final product with consumer before launching. Okay, so a product. Okay, so consumer, that's mean you use the product as directed in their home environment and answer the question. Okay, so to capture the consumer's impressions and experiences. Okay, so the product appeal and purchase intent are two key parameters that predict the consumer acceptability of the product. So that means we can conduct, okay, the questionnaire to gain the overall satisfaction of consumer about the product and assessment of the product attribute, so as we can further improve, okay, so and ensure the product acceptance. So that's why many protocols will be used. Okay, so so to test on the safety and effectiveness before marketing. Okay, before putting launching to the market. Okay, so for example, we have the efficacy testing, as I told you. Efficacy testing can be conducted as blind test, double blind, or open label test. So, and then we did what we call the expert clinical grading plus objective instrumental bioengineering method that is used to evaluate the skin status and facilitate statistical comparison. Okay. So here are the examples of the scales for the expert clinical grading. Okay, so it is a numeric scale. Okay, so usually used to dimensionalize the visual assessment. Okay, of course, this will be conducted by an expert. Okay, so and then we have some what we call the bioengineering method that we use the sophisticated instrumentation and procedure to make sensitive and reproducible measurement. Okay, so under different factors to assess the skin condition and determine changes with product usage. Here are some examples of bioinstrumental method. So for example, we want to measure the skin hydration content. Okay, so the theory behind is the change of electrical capacitance or resistance, okay, from the instrument, or so we can measure the TEWL, transepidermal water loss content, okay, using evaporometer, okay. So, and then we use the thermal top meters, ballastometer, and custometer to evaluate the elasticity, okay, and firmness of your skin, okay. So, and also the IR device to assess the skin temperature. So, and then we use the chromometer to evaluate the skin wetness or the darkness. And finally, so we can use the skin oilers and the presence of flake. So will be used. together with the adhesive tapes, okay? So to evaluate the oil content or sebum content. So that's why if you want to have the imaging, okay, so you can use the confocal microscope or optical coherent topology, okay. So that is some advanced microscope, so to see the surface texture or morphology, so of the skin structure, okay. So and finally, we may use the image analysis, that is using the computer. So for video microscopic image, okay, so that is taken under normal polarized fluorescent UV light condition. Okay, and then we can have the fringe or shadow projection technique. Okay, so to assess, okay, the skin topology. Okay, so whether especially, okay, so for the sunlight intensity. So sometimes if you want to see deeply into our skin, whether it has some disorder, okay, so or any change, a structural change, so of your epidermis, okay, or even the dermis, okay, layer, then you need to have a skin biopsies, okay. So all of these methods to be used it quantitatively, okay, sensitively, and reproducibility. So to enhance, okay, so the testing options, okay. So this, okay, so together with the all types of testing, okay, so can be used to evaluate, okay, so the stability, safety, and toxicological information of your final formulation. Okay, so finally, we will send the questionnaire can be used as the clinical study design. Okay, so okay, so you in the questionnaire you can ask the subjective irritation parameters. Okay, so to evaluate the consumer by evaluating the consumer perception. Okay. So after all these long testing step, everything is okay, then you may scale up the process. Okay. So to begins with a series of lab batch, so leading to mid-sized pilot, okay, or full-scale production batch. Okay. So, and then you may have some trial production batches, okay? So before commercialization, okay? So, and stability testing is used to determine the actual processing condition. So that may have impact on the final product, okay? So, all these manufacturing step, so should be validated according to the current GMP, good manufacturing practices, to ensure the quality has been controlled and assured. So another topic, okay, that is about the control of the Chinese medicine in Hong Kong. Okay, so it is under the jurisdiction of Chinese Medicine Division under the Department of Health. Okay, and they are responsible for the enforcement of Chinese medicine ordinance. Okay, so and these ordinance provide, okay, for the regulations about the practice of Chinese medicine practitioner, the use, manufacture, and trading of Chinese medicine. Another statutory body, we call it as the Chinese Medicine Council of Hong Kong. Okay, so that is used to implement the licensing system of Chinese medicine trader and registration system of proprietary Chinese medicines. Okay, so that's why the safety, efficacy, and quality of PCM, okay, will be assessed. Okay, including the distribution, storage, and labeling of Chinese herbal medicine will also be regulated. Okay, so and that's why the Chinese Medicine Division provides some professional and administrative support to the council. Okay, so and also serve as the public health functions. Okay, so in terms of the development of the Chinese medicine in Hong Kong, so it's a kind of systems of registration, licensing, and labeling. Okay, to regulate the manufacturing, distribution, retail, and import, and even the export of the Chinese medicine. So that's why we have the Chinese Medicine Development Committee that gives us recommendation to government about the direction and long-term strategies. So for the development of the Chinese medicine in Hong Kong. So how about testing of the active ingredient in the herbal medicine that is conducted by government Chinese Medicine Testing Institute? And then we have the Hong Kong Chinese Material Medical Standards Office. So that is to manage and coordinate the Hong Kong Chinese Material Medical Standards. Okay. So you can find the information from their website. Okay. So, and then to enhance the quality management of the PCM. Okay. So in Hong Kong, that is applied to Chinese Medicine Board formed under the Chinese Medicine Council of Hong Kong, and that applied to certificate for manufacturer. So they must obtain the GMP standard in order to manufacture, okay, so the PCM in Hong Kong. So the board prepared the Hong Kong Good Manufacturing Practice Guidelines for proprietary Chinese medicine, as I said to you. So that covers the fundamental items of GMP in the aspect of PCM and to promote the standardization of PCM and to assess the quality and safety of PCM and safeguard the public health and increase the public confidence in using the PCM in Hong Kong. The last part of this chapter is about some current development of cosmetic and personal care products. So recently, okay, we usually heard about the nanotechnology applied to cosmetic products. So one very common chemical that we use is what we call the silver. Okay, silver can be used to kill the bacteria and fungi. Okay, so by using the nanotechnology, so we make the size smaller. Okay, so to make it easily penetrate deep layer of our skin. So that's why we use the silver colloid. That is a neutral aqueous solution. Okay. You know that the skin is semi-permeable, okay, that is not allowed even, okay, the nanoparticle to pass through easily, okay? So unless our skin barrier is disrupted, okay? So the evidence shows that if it is 20 to 200 nanometer, cannot penetrate the skin barrier easily. But if we can make it smaller than 10 nanometer, it can reach the deeper layer of structural corneum. Okay, so that's why the silver nanoparticle could provide a safer alternative to conventional antimicrobial agent. So in form of topical antimicrobial formulation. Okay. So the use of the phyto-based cosmeceutical, okay, so a lower penetration and high compound instability, okay, so that's why nano sizing of these compounds, okay, enhance the aseptic field in different cosmeceutical products, okay. So, and this phytocompound is good for some parameter, for example, photoaging, inflammation, hair loss, lip care, psoriasis, and UV toxicity. So, and how about the dosage form? Okay, so of nano sizing. So we use either nano emulsion. Okay, so the picture depicted on the upper up. Okay, so here. Okay, so that is nano emulsion, and then the liposome. Liposome, in fact, is the lipid bilayer. Okay, so lipid, just like this diagram, lipid bilayer that enclose aqueous core. So the liposome is rather a stable dosage form. Okay, so for delivery of active ingredient. Okay, in the cosmetic product. So, and the next will be what we call the solid lipid nanoparticle or carrier, or even the hydrogel. Hydrogel, okay, most popularly used in the mask, okay, because hydrogel, so like this diagram, so can trap, okay, So the active ingredient, it's just like a mesh, okay? So that can be used to trap the active ingredient, okay? So better. So let's introduce some nano product to you. So for example, we have nano anti-aging product. So you know that the skin collagen loses its physical appearance. So in many ways, for example, sebum production decrease, sebum production decrease, dry skin, texture loss, age spot, or no skin. Okay, so that's why in order to have the anti-aging effect, we should have anti-oxidative mechanism. So you can combine the use of polyphenol and retinol, okay, nano emulsion, okay, so with nanoparticles, okay, or some what we call the leaf flavonoid, okay, or the encapsulation of resveratrol and curcumin, okay, so, and a nano moisturizer. So skin dehydration can be prevented by applying moisturizer. So which can enhance our skin flexibility and maintain the moisture content. Okay. So that's why the moisture preventing barrier on the skin, okay, is the strut corneum. Okay. So dehydration results if there is loss of skin from this layer. Okay, so that's why applying moisturizer create a thin film on the outer surface of skin. Okay, so that's why the use of nano emulsion, nano liposome, solid lipid nano carrier. So for moisturizer with low viscosity and non-creasing effect. Okay, so these are the below will be some example of nano moisturizer. So another type will be the nano cleanser. Okay, so that is a thin hydrolipid film covers the skin from decomposition product of keratinocyte and secretion of spacious gland. Okay, so the film on the skin surface may attract the undesirable microorganism. That is the bad side. Okay, so that's why skin cleansing directly promote the removal of skin surface bacteria directly. So and then nano cleanser. So the cleansing activity reduce the pore size and skin sebum content. So we will use the liposome phyto compound, okay, or even the neosome, okay, with the combination of the naruric acid, curcumin, that will be used to reduce the acne caused skin infection, okay. And then nano screen. Okay, so there are two types of screen, of course, that is the physical sunscreen. So we may use the nano sized zinc oxide or titanium oxide. So that is stick on the surface to reflect or block the sunlight. Of course, we still have the chemical sunscreen as is the organic filter that is used to absorb the UV light. Okay. However, chemical sunscreen are more irritating to especially sensitive skin. So these are the example. So and then for nano screen, so product, okay. So you may take a look at the SPF value. I think of you know that is the fraction of sunburn producing UV rays that reach the skin. Okay. So what means by SPF 15, for example, that means 1/15 of burning radiation will reach our skin. Okay. So that's why the sunscreen efficacy can be determined by multiplying SPF, so by the length of time to suffer a burn without sunscreen. Okay. So these are the example. I will not take a look at one by one. Okay. The example of nano screen. So and then the last part will be the nano hair care. Okay. So hair care is a hot topic nowadays. Okay, so let's talk about why, especially for male, the hair loss are related to scalp inflammation and growth disorder. Okay, so now our hair consists of three major parts. Okay, the outer layer is what we call the cuticle. Okay, so the middle layer is what we call the cortex. Okay, so and the inner layer is the medulla. Okay, so for the skincare product, it only affects the cuticle layer and cortex layer, no effect on the medulla. Okay, so that's why improper use of tools, excessive heating, okay, so and then using of chemical, so may damage the outer layer cuticle. Okay, so cortex has two types of melanin. Okay, so that give rise to our hair color. Okay, so chemical hair relaxing, thermal styling, red setting, and hair coloring oxidation, so will cause temporary or even permanent change to the cortex layer. So that if excessive con or if there is continuous process of this, so that will cause a permanent damage to the cortex layer. So, okay, so the skin hair product will not affect the function of medulla. Okay, so the general causes of hair loss may be attributed to getting too much vitamin A, insufficient protein intake. Okay, so the change during pregnancy, the genetic factor, animal, okay, hypothyroidism, so lost weight very quickly and undergo some chemotherapy or under some stress. Okay, so you will have a hair loss. So that's why you can use the rice and obstacles. So I used it to enhance the hair growth, color, and appearance and protect our hair from UV-induced degradation. Okay, so and then the pomegranate hydro ethanolic extract, okay, was used to reduce, okay, the hair follicle wet fading effect and enhance the skin, the hair color. Okay. So finally, we can use the nano hair care product. For example, you can use the curcumin-based liposome or some complex, okay, or combination. So that enhance, okay, so the penetration of the hair follicle and non-follicle cell pathway. Okay. Now, these are the some common or current contemporary development of the cosmetic product. Of course, there are still many. Okay, so that's all about the chapter 3 of these modules. So thank you very much.